Hello, I'm Gail Humble. I have a genetics background, and then I have an aesthetic and regenerative practice in San Francisco and Los Angeles. A little background on me. I did 5 years of research on the aging face. I did the Sculptra FDA trials, and I've been advisory for a lot of the big aesthetic companies. I first read Dr. Kruger's paper almost 1 year after he wrote it. I read it. I went to myself, wow, what's going to happen? Everything's going to change. And nothing changed at all. And that's actually how long I've been working with Klotho on a topical level. Being in the superficial skincare form, we've developed a lot of things and done 3 clinical trials, 3 cellular trials. All are IRB approved and published, except for one that's being written up right now. And we're going to talk a little bit about our studies, but more importantly, I'm going to go over and be completely transparent about our process and how we harvest the Klotho protein from mammalian cells, the concentrations we use, and what our results have been, which have been more than impressive. One thing I want to say, I love the joke about when you don't have a kidney or a dialysis patient, when you don't have a heart, when you're dead. But if you don't have your skin, you're just funny looking. Disclosures, I'm CEO of Klotho Skin. This was incorporated 3 years ago. And also I'm co-founder of Klotho Longevity, which I've started with Joe Cleaver. And we can talk a little bit about this afterwards, but the attempt is to get a middle range product of exogenous Klotho protein that will raise your endogenous blood level that the wide use public can have access to. All right, the skin may seem rather superficial, but I want you to know it has its important functions. It serves as a barrier. It protects you against injury because of sensory sensations. There's 30 to 40,000 individuals skin cells shed per hour, home to over 1,000 bacteria species. And of course, it's very important when it comes to vitamin D protection. Your skin is the largest organ, and it basically renews every 21 to 28 days. Everyone knows there's different thicknesses in different areas in your body, and this reflects in the cellular renewal. Your skin is a result of aging because of 3 main things: oxidative damage, your genetics, and repetitive facial movements. We know the UVA, UVB, and UVC are so important to protect for skin health. So often in Western medicine, I think we wait until there's a problem and then we throw a drug at it or a cure. But this is true with skin conditions as well. We've got a ton of skin products, half of which don't work. I don't know, for the men and the women in the audience, I'm sure you all have a drawer full of products that are no longer working that you spent money on. What if we can change this? What if instead of waiting till we're treating aged skin with photo damage and UV damage, what if we can slow the way your skin ages? And what if we can delay age-relating disease? That's where the Klotho protein comes in. So we all know there's a UVA, a UVB, and a UVC now. There was a big release in the news about 2 months ago that, oh, wow, sunblocks are not the holy panacea that we thought they were. As a matter of fact, your chemical sunblocks don't even cover between 320 and 400 nanometer range. Your physical blocks are best, but in actuality, in order to have them be efficacious, you have to apply them every 2 hours or more frequently. So we really need some other help, since this is one of the main 3 reasons our skin is aging. We have to protect ourselves against UVA, UVB, and now UVC. Okay, we may not be as important as the heart and kidney, but there's about 70,000 people per year that die from non-melanoma skin cancers and about 60,000 that die from melanoma skin cancers. The total is small. It's only 128,000. But what is not small is the projected $270 billion that is spent or is going to be spent just in the skincare market. I'm not going to stay on the slide except for to confirm what I always wondered. Klotho, the Greek fate, is spelled with a C. And I did ask Dr. Kuro yesterday if it was changed to a K as his only recognition of finding this important protein. And that is true. We know Klotho does a lot of things to the body. And we know my slideshow is stuck. Can you advance it? Oh, great. Okay, we know the Klotho protein. We know when the gene's upregulated, we get the Klotho protein. We're monitoring the Klotho protein in the blood, the CSF, and the urine. We know all the wonderful things it does for the body, but let's talk a little bit about what it does for the skin. It actually delays the aging of your skin by blockadating oxidative stress. We know this is by increasing FOXO1, increasing superoxide dismutase and catalase. It actually increases nitric oxide, which allows vasodilatation and more oxidation of the tissue. It decreases transepidermal water loss. We are going to find out that at the right concentration, it actually increases two other genes that are known to preserve the end of telomeres, and these are the holy grail of aging. We're going to find out it actually protects us from UVA and UVB damage, delays the aging and the age-related diseases, actually reverses the skin atrophy or the skin thinning of the skin as we get older, activates platelet factors, so it's actually acting as an internal PRP, and acts as an anti-inflammatory. Now that's a long list for any one product to say that it can do for your skin. This is showing what it's doing on a cellular level. We've all talked on that, and there's one other thing I want to mention that I didn't hear mentioned a lot. Klotho has a diurnal variation as well as a U-shaped curve. But on a cellular level, one of the most important things is when you have a higher Klotho level, you're turning on your FOXO1. Now, everyone's talking about longevity nowadays. And if you ask AI, what is the most important longevity gene? You're going to see FOXO3 and a bunch of other genes. And that's all very exciting. But my point of view is we don't really want longevity without anti-aging. No one wants to be stuck at 90 years old for 30 years. So when your Klotho age suppressor gene is increased, you increase something known as FOXO1. This then in turn increases superoxide dismutase and catalase. This is more than an antioxidant. This blocks oxidative stress. And on a cellular level, especially an organ as big as the skin, it's very important. This is showing how it's working on the insulin pathway, blocking the autophosphorylation, increasing FOXO1. Okay, now this is when the talk gets exciting. This you get to know exactly what our company has done. This is how we upregulated the Klotho gene in adult mesenchymal stem cells from fat, and we got the Klotho protein at a reliable, reproducible concentration. What you're looking at here is called an open reading frame, and I want to point out Sarosh Vesak, my chief medical officer in the front row. If you have any terribly complicated questions, I'm going to refer you to him on this. He was very important to all this. So this is a construct. It's a DNA engineered fragment, generally small. And what we did was we used both a plasmid and a lentivector to act as an arrow for those that are not cell biologists. And we basically shot this on and on switch into the DNA of adult mesenchymal stem cells, meaning they're from fat, in a petri dish. When we did this, we were able to upregulate the Klotho gene in these cells. Now, if anyone's ever worked with stem cells, you know they're a little difficult. They're like teenagers. They want to grow up and become doctors and lawyers. They don't want to stay as adolescents. Or else, if they're genetically modified, they want to kick out the genetic modification or they want to die. So, to do a genetic modification to get enough regulation of the Klotho gene and therefore get the Klotho protein in the cell conditioned medium, which we'll talk about, it's very difficult. This was our first patent. I will add that we've gotten 3 patents. The first one is a complete failure. I'm happy to discuss with you if you guys want to know. The second one worked where we caused the genetic modification, the upregulation of the Klotho gene to get the Klotho protein in the cell conditioned medium. And the third patent, which we're going to be working on, is where we doubly transfected these cells with not only the Klotho construct, but H-TERT. So we basically immortalized our cell line. The reason we did that is we needed to reach scalability. So we know the cells are hard to genetically alter, then we have to multiply them. The longer they live, the more passages they go through, the more cell conditioned medium we can collect. After we collect the cell conditioned medium, of course, an ELISA assay is done to show the concentration of the Klotho protein in the cell conditioned medium. And this is our final ELISA assay. Okay, I told you earlier we did 3 cellular studies. I'm going to go over each one of them. The first one is probably the most exciting. This was actually done by Peter Sikora at Amelia Labs. I told you I've been working on this a really long time. What I didn't tell you is people have been walking away from me for years when I would try and talk about Klotho because nobody knew what I was talking about. So we used to go to these medical conferences and nobody would come by our booth. Well, Peter was, I think he has 3 PhDs, and he came right over to us and he said, Gail, I just got an NIH grant for a new alkaline comet test that we're going to talk about. And I'd like to take your cell conditioned medium and see what it does to UVA strand damage. You heard me talk with Joe Cleaver and say, I think strand damage can, in certain cases, be reversed. So this test, what it actually does is it monitors the DNA damage. You basically take cells, hit them with a high concentration saline solution and detergent, make nucleotides, break these apart, and then use electrophoresis to either see the intact strand or the smaller strands. This makes a comma, and that's a comet, and that's why it is called a comet assay. So what Peter did was he took 2 sets of fibroblasts. One he put in a control, and the other one he did in our cell conditioned medium with the Klotho protein and second generation growth factors. Because what we found out, in addition to the Klotho protein, we actually had quantitatively and qualitatively greater growth factors than that comes from normal cell conditioned medium. Okay, so you got the two groups. One's in control, one's in our cell conditioned medium. And then he did UVA radiation on these cells. Some of these cells died. Well, Peter called me like at 5 o'clock in the morning. I think he's on the East Coast. And he was so excited. And he said, I know I can't reproduce it, but we have 40% reduction in DNA strand damage. Well, he did reproduce it. He did the study 15 times on more than 250,000 cells. And this study was already published. So we were pretty excited, but he still went further. He said, let's reduce the UVA radiation equal to just 8 hours of sunlight. So there is no cell death. Same thing happened. 40% reduction in DNA strand damage. Now, this is what's causing tumors. We can't say it's tumor suppressive. We can't make claims, but this is really important knowledge. And this is showing that Klotho has a clear protective response against oxidative DNA damage. We know DNA damage is critical for tumor formation. And when it's unchecked, that's resulting in both skin cancers and part of my feeling as to why we're getting cancers or why Klotho is tumor suppressive. Okay, we did two other cellular studies. The one on the right is almost an identical study, only this time we took the fibroblasts, the control, and the ones in the cell conditioned medium, and we used UVB irradiation. Now, this is really important because this breaks down your collagen and your elastin. This results in photo damage. We found that when we had ours in the correct concentration, the cell conditioned medium, we actually had an increase in collagen 4. Collagen 4 is not talked about that much, but it's really important in your basement or your basal membrane. This is maintaining the structure in your skin. Oops, hold on. Uh-oh. Can you go back? Oh, technology is not my friend, just in case you didn't know that. Okay, our other cellular study was we decided to monitor other genes at different concentration of the Klotho protein. At the right concentration, actually 1%, we found that there was actually an increase in two other genes. That is HTERT and SIRT6. So these genes serve to preserve the caps, so the telomeres, the holy grail of aging. You know, it's a certain length. Some people think we can elongate them, some people don't. But at any rate, as it continues to have the tips broken and get shorter and shorter, it finally reaches a certain level. It's too short, and the cell dies. Then these cells go on and either are destroyed or they become senescent or zombie cells that you also heard Dr. Cleaver talk about. Okay, let's go into our human clinical trials. First of all, this is how we make our product. We have 3 SKUs. This is not marketing. I'm just telling you what's in each one. We have a face serum, which is 25% cell conditioned medium and other ingredients. Doesn't have perfumes. We spent about a year working on the fragrance because it's not a pleasant one, but now it's a very luxurious product. This is your big gangbusters. It says 25% cell conditioned medium. We also have an eye serum with 15, a night cream with 10%, and we just launched our microneedling solution. This is how we make it. We combine fibroblast cell conditioned medium with our genetically altered adult mesenchymal cell conditioned medium. Some of the fibroblast cell conditioned medium have earlier onset, and that's one reason we did it. But usually you keep your permanent changes by about 3 months. Okay, full transparency. What's in the goop and what's not in the goop? Everyone knows the Klotho protein is large, 120 to 130 kilodaltons. So how do we even know it's getting through the skin? Well, it may not have to actually do its work similar to the blood-brain barrier, but we actually use liposome technology to cloak the protein and fool the skin. We then have second generation growth factors that again are quantitatively and qualitatively greater than plain stem cells, cosmetic ingredients. But what's not in the goop? So often I hear about salmon stem cells, snail, plant growth factors. The reality is probably most of those, they may not do any harm, but you're going to have human growth factors if you want to treat a mammal, or mammalian growth factors if you want to treat a mammal. So there are no endotoxins. Every group is checked for hepatitis, HIV, HPV, bacteria, fungus, and mycobacterium. Okay, the first study was published in Skin and Aging. This was done in my office in San Francisco. We had 10 patients. It was our first study. We asked nothing of them other than to not change their life experience at all and use the face serum morning and night for 3 months. We evaluated them monthly. We had a patient questionnaire, and then we evaluated them with a Visioscan. A Visioscan is an objective way of monitoring photo damage, texture change, and rhytids or wrinkles by using a white light. It monitors you 1 out of 100. So if you start at 40 out of 100 for your age group and you end at 60, that's a 20% improvement. The results were pretty impressive. Mind you, they did nothing except for add a face serum, one pump in the morning, one pump at night. Um, we found that there was a 30% reduction in rhytids. Sorry, I think this converted from a PDF as well. So there's some typing not coming out. A 31% improvement in texture and a 31% reduction in photo damage. So this was published and we were very excited about it, but we knew we had to do a study somewhere else where we weren't that involved. And Pearl Grimes is a KOL. She's in Beverly Hills. She's a well-respected dermatologist, does a lot of work with vitiligo. Our study on this came out in the Journal of Dermatologic Drugs a week ago, and I was excited to find out that people had read it here. So this was a double-blinded study, so it involved 20 patients. 16 patients had the Klotho our skin, Klotho Skin, morning and night, and then we had to make a placebo that was exactly identical with the serum, the smell, the boxing, everything. Nobody knew, either the patients or the doctors, as it was double-blinded. Again, the study went for 3 months. They applied it morning and night. Visia scans were used as a follow-up, as well as a mexameter, which is a way to monitor the amount of melanin in the skin. The results were impressive. Overall, there was a 2.5 times improvement. What we now call this is the Klotho Glow. And as we go through some of the photos going forward, I want you to notice the Visia scan is completely fixed and the lighting is always the same. But in each of our patients, in the second picture, it looks like there's a brighter light on them. And that's literally the global improvement. There was also a 36.5% reduction in photo damage, 18% reduction in melanin, 11% reduction in erythema, 38.5% reduction in dyschromia, and 27.5% reduction in rhytids. It makes sense that this is going to work on rosacea patients because it's also an anti-inflammatory. We then had 4 patients that volunteered to go ahead and have biopsies, which I always found amazing, but yes, they're willing to do it. This is one of the biopsies showing the decrease in melanocytes in the dermal layer, an increase in the collagen, which was hugely significant. That's the darker pink. And finally, the increase in collagen and elastin. What are the patients at baseline and week 12? In this case, you see the reduction in erythema. Again, this is just using the face serum morning and night. Well, so that one came out, and now we're writing up our third IRB approved clinical trial. This was done by Steve Yolen right here in Newport. He had 16 patients, and the only thing he did different was he had them use the eye, the face, and the night for 4 months. And he had them use the night, morning and night. And I think that actually is increasing the penetration a little bit. The results were amazing. At 4 months, there was a 50% reduction in photo damage, 68% reduction in dryness, 42% reduction in erythema, 38% reduction in dyschromia, 32% reduction in rhytids, and this is the most important one. It's a 10% reduction in atrophy. We all know the skin's thinning as we age. This is actually starting to reverse the process. This is a patient perception chart. I want you to notice it starts at 88%. All the patients were 92% or higher. 50% of the patients were at 100% satisfaction. As we go through each one of these photos, I want you to notice again the brighter light on the second photo. We did not have exclusion criteria. I would have not let the second patient in because of the photo damage was so severe. But again, she almost looks like she's had a laser resurfacing. We did find as we go through these, as the patients got younger, they did respond faster. Okay, as of last December, we launched our microneedling solution. This is a 50% cell condition medium. The only other thing in this product is a small amount of water, a micronized HA. The reason the micronized HA is it allows 95% penetration through the skin in 24 hours, and that's intact skin, so it's going to help with the penetration. It also acts as an anti-inflammatory equal to dexamethasone, so you can use it after radiofrequency devices, and it actually decreases your immediate erythema and helps with the healing process. It does not need to be refrigerated, which makes it a little easier to work with than exosomes, and each one is filled to 6 ml. We are going to be starting our studies, and hopefully we're going to be working with Howard and his face mask with this as well, because I think when we put them all together, we're going to have even more amazing results. I've got to present this case of one. I had a colleague who was kind enough to offer to microneedle with the Klotho Complex microneedling, the actual cell conditioned medium. And this person monitored their senescent cell load before and then afterwards, and then 24 hours afterwards. So it does have a systemic effect in that the senescent cell load was reportedly reduced by 2/3. Okay, everyone knows about growth factors, PRP, exosomes, and Klothos. I am going to say my favorite little thing here, exosomes, or as you call them, yeah, you're correct. They are no more than envelopes. Your exosome is good as where it came from. So the source becomes very important. But assuming it's coming from a young source, they're all great in cell communication. They're all great in different things to help cellular maintenance, but only the Klotho is preserving the end of the telomeres, which is slowing the aging of your cell. So it's slowing the aging of your cell, and then it's actually helping your cells replicate more accurately by decreasing DNA damage and acting as DNA protective. It also blocks oxidative stress, increases nitric oxide, acts as an anti-inflammatory, and acts as a platelet activator. So we know the population is aging and the skin is the largest organ. It is important. Vanity is important. It's one thing that keeps us wanting to regenerate our bodies. I do believe one of our biggest problems, and this was said by Dr. Abrams in her paper, we need to have a standardized blood test. Dr. Cleaver touched on it. Some of the tests are in nanograms, some of them are in picograms, and we need to know that we're not just following trends, but we're actually looking at real levels. Also, one final thing I wrote at the bottom, could the skin ever be considered for a delivery form for Klotho? Okay, eyes up, everyone. This is a lot of work. We're going to see if this works. This is basically what everyone wants. A 300-year-old mind with a 30-year-old body. If you guys have any questions, you can always reach out to me at gail.humble at Klotho Skin, or here's our website. That's the office number, or you can just tap me on the shoulder. And I think we're ready for the next speaker. You guys want to see a 90? Do you guys want to see a 90-second video that makes, okay, go ahead. It's a little markety, but it's not for that, but it's fun how we put science into a cartoon.
What if your skin didn't have to age like everyone else's? Introducing an innovative new skincare line that can help you realize that exact possibility. In 1997, the Klotho gene was identified as the first documented age-suppressing gene in mammals that can delay aging when overexpressed. And now, Klotho Skin has patented a process that genetically modifies human stem cells to overproduce the Klotho gene and collect the Klotho protein. Combined with second-generation growth factors, this cell-conditioned medium is the main ingredient in our revolutionary patented face and eye serum. Through further testing, we found that exposing cells to the Klotho protein upregulated two other genes that prolong cellular life. Not only is Klotho the first and only skincare line which works with genetically enhanced cells, but studies show that greater than 95% of users noticed improvements in their wrinkles, texture, and UV spots, demonstrating that the Klotho protein may be regenerative, protective, and preventative. Klotho is the future of skincare, an all-in-one treatment offered in medical offices and spas to address skin photoaging. For more information on how your skin does not have to age like everyone else's, visit us online at klothoskin.com.